Our next speaker is Alex Williams from Stanford University. His field is theoretical neuroscience, and his advisor is Surya Ganguly. And he did his practicum at Sandia National Laboratories at California in 2016.
All right, thanks. Everybody hear me? Great. I want to start by thanking the Krell Institute and all the organizers of this fellowship. I'm really grateful for all the support and scientific interactions that I've had here over the last 4 or 5 years. And I'm just very excited to be up here talking about my research. So I'm going to tell you about a couple collaborations I've had with a few neuroscience research groups at Stanford and other institutions. And this work will sort of draw on a mix of statistical modeling and large-scale computing frameworks, but I actually think the most interesting part of the talk for an audience like this is the new scientific questions and data sets and applications that we're now sort of able to get a foothold into. So the motivation behind this work, broadly speaking, kind of goes back to this trend that dates back to the 1960s, which is a doubling or an exponential growth in the scale of neural recording technologies. Roughly every 20 years, the number of neurons that we can record from simultaneously from the brain increases tenfold. And this has been called by various people the Moore's law of neural recording technologies or neurosciences. It's not growing quite as fast as the original Moore's law, but is nonetheless a pretty remarkable and rapid expansion in the size of the data sets that neuroscientists are able to collect. And just on a practical level, going back 20 years, the typical neuroscience lab would be able to record from maybe 10 to 20 neurons at once. And today, we're able to record from multiple hundreds of neurons at once, if not thousands of neurons at once. And to be completely frank, I think the field wasn't fully prepared for this sort of qualitative shift in the scale of the data sets that we're able to collect. So there's lots of people, including myself, that are kind of scrambling to play catch up and keep pace with this rapid advancement. So just to give you a sense of what these data actually look like, this is the record in the published literature in terms of a scale of neural recording. This is a recording from 3,000 neurons simultaneously from a mouse brain. This is published earlier this year by Carson Stringer and colleagues from Genelia and University College London. What you're seeing here is a raster plot where each black dot denotes a single spike or action potential from a neuron, and these are the electrical impulses that are sort of the basic unit of computation and information processing in the brain. So this is here a roughly 15-minute recording. The horizontal position is showing time. The vertical position is showing you which of the 3,000 neurons is firing that spike. So we have this really large multidimensional time series. And if we zoom in on a much smaller window, now I'm showing you a 3.5-second window of this same data set, you can see that there's also a lot of fine-scale structure and oscillation within this data that is obscured when you look at this larger view. So I think it's a real challenge for statistical neuroscience to build understanding across these timescales and help scientists understand the sort of complete picture here. I want to give one more quick example just to drive home the disparate timescales that are typically at play in these data. So this is a different study, a slightly older study by Andy Peters, where he recorded from motor cortex in mice learning a very simple lever pressing task. And he was able to record from the same animal over the course of a month, roughly. And what I'm showing you here is 3 different recording sessions as the mouse progressed from a novice to an expert at this task. And as the mouse becomes adapted to the behavioral task, its motor movements become more stereotyped and repeatable. And as this happens, the neural activity you can see becomes also a lot more structured and precisely timed to each individual lever press. So the red arrows here are showing you the time of the lever press, and the dots, again, are showing you action potentials. So the details of the study don't matter so much, but what I want to sort of drive home is the sort of incredible capability that we have to record both sub-second, really millisecond level structure in these data that can encode individual actions. But it isn't until we zoom out and look at the same data set over multiple days or weeks that we are able to appreciate these striking effects of learning, for example. I don't think this is a statistical, from the standpoint of statistics, we haven't really agreed on the best methods for pulling out effects like this in an unbiased and unsupervised manner and quantifying them. So this is going to be one of the themes of the research I show you. So one of the things I proposed in my PhD, I mean, I guess it's not necessarily a new idea, but it's one that I think definitely should be more broadly appreciated within the field of neuroscience, is how to represent your data. And the sort of simplest way to represent these high-dimensional neural recordings is as a three-dimensional data array, or what's known in some fields of mathematics as a third-order tensor. So the idea is that each time the mouse presses a lever, you get some series of action potentials across many hundreds or thousands of neurons at once. And the mouse repeats this behavior over many trials. And you can collect all of the matrices you get and put it into a three-dimensional data array, which has dimensions, numbers of neurons that you're simultaneously recording from, number of time bins, discretized time bins in each trial by number of trials. And this can be a moderately large data set. So if you have 1,000 neurons, 1,000 time bins, 1,000 trials, this ends up being 1 billion independent elements or data points in this data array. This isn't quite as big as some other data arrays that you might encounter in simulations of nuclear reactors or things of this nature. But one of the things that makes this problem particularly challenging for us is that we don't have that underlying physics or a forward model telling us or informing us what the structure in this data set should really be. We're still at a pretty early exploratory data analysis phase in most cases. Okay, so what we'd like to do is measure or estimate the firing rate of individual neurons over multiple trials. And in the simplest case, if the firing rate was exactly the same, there wasn't any, it was an entirely stationary process, there wasn't any learning over trials or changes in cognitive state of the animal, then a reasonable thing to do would just be to average the spikes we see on each individual trial and then just be done with it. But as I've shown you already, there's many cases, scientifically interesting cases, where the firing rate or the temporal fluctuations in firing rate are changing as the animal is learning a task. And the purpose of this slide is just to say that this is a really hard problem from a statistical standpoint once you start to relax these assumptions. So this is a paper from Amarasingam et al., which basically shows that if you let the neuron's firing rate vary arbitrarily from trial to trial, then the problem is more or less hopeless in some sense. A statistician would call it non-identifiable. So this slide is showing you 3 different models, and each one of these models is equally consistent with the data set that we observe at the bottom. So the idea is that we can't let the firing rate change arbitrarily from trial to trial. We have to make some sort of simplifying assumption about the system. So my work has sort of come at it from the other end, which is, what are the maybe simplest assumptions about the system that we can make? And I won't go into it, but these assumptions do have some sort of precedent within the experimental neuroscience literature. And I had a couple different projects during my PhD, but the 2 main ones that I'll summarize for you today can be summarized in these 2 schematics. So the first idea is that we're going to look for groups of neurons that are show correlated modulations in their amplitude on a trial-by-trial basis. So these are 3 neurons that are either enhanced or suppressed together as a unit on a trial-by-trial basis. And then the second part of the talk, I'll talk about changes in the timing or the latency or duration of these dynamics. And again, we'll sort of assume that there's multiple neurons that are showing these trial-to-trial variations in a correlated manner. And the real, scientific meat of what I did was to show that these simple hypotheses could be formalized into statistical models, and furthermore, that they could be scaled to these really large data sets in a computationally efficient manner. So in the first part of the talk, I'll just very briefly tell you about a tensor decomposition model that formalizes this idea of amplitude modulation. And in the second part of the talk, I'll talk about curve registration or time warping methods that can formalize this second form of variability. So because of time constraints, I'm just going to give you a very simple taste of both of these methods, and I'll leave plenty of time for questions, hopefully, so that we can go into more details if there's interest. Okay, so the idea behind tensor decomposition is actually a relatively straightforward generalization of a popular technique known as principal components analysis, or PCA. The idea here is that you want to find a small number of component vectors that describe a matrix of data. And the idea behind tensor decomposition is relatively straightforward, that rather than approximating this data as a sum of rank 1 vector outer products, we'll summarize it as, again, rank 1 vector outer products, but now generalizing the outer product to contain a triplet of vectors rather than just 2 vectors. So this is the mathematics of the problem, but intuitively what this model picks out are low-dimensional embeddings of the neurons, so groups of neurons that fire together. All these neurons have the same temporal firing pattern within each trial, and then across trials, their amplitude is modulated by this third low-dimensional factor. And this was work that I actually started at my practicum at Sandia Labs with Tammy Kolda, who's an expert at fitting these sort of models. And we published a paper together that went through various applications of interest to neuroscientists using this model. I won't go too far into the details other than to say that we can pull out in real data sets groups of neurons that tend to fire together. And then in this particular primate that was learning a motor task, this group of neurons was basically amplitude 0 at the beginning of the experiment. But then after the animal began to learn the task, it slowly increased in amplitude. So this was able to extract out a useful feature of this data set that we really didn't have like a handle on before using this method. Okay, so very briefly, just talking about this time warping model, note here that, okay, so I'll remind you that the time warping is going to be looking for changes in the timing or duration of dynamics, and this is a type of trial-to-trial variability that is explicitly not accounted for in the tensor decomposition model, or it's at least not accounted for in an efficient manner. This can lead to a high-rank structure in your data. So just to motivate this method, I want to show you that small amounts of trial-to-trial variability and timing can obscure really striking features in your data. So here I'm showing you a single neuron over 1,000 trials. And there's other neurons in this data set, but for simplicity, I'm just showing you one. And I'll tell you that there is variability in this data set on the time scale of about 1/10 of a second. So I want you to just look at this data and see if you can pull out what the underlying pattern might be, given that I've told you that there's some trial-to-trial variability. And then what I'll show you on the next slide is the same exact data. And the only difference is that I'm going to reorder the trials based on the time warping model. And when I do this, I hope that this pulls out a pattern in the data that we could have never really seen in the raw data, which is this about 7 Hertz oscillation in the spike times. And the reason this is obscured in the raw data is that there's a decent amount of temporal variability or jitter in the data, which totally obscures the fact that there's an underlying oscillation here. So again, these are the sort of features that are buried in this high-dimensional data set that aren't necessarily, or actually have been overlooked in this particular lab. They had no idea that there was an oscillation here. Just very quickly, the way that we fit this model is for each neuron, we try to fit a firing rate function, and then we modulate that firing rate function on a trial-by-trial basis using what's called a warping function, which more or less just re-indexes the time axis of this response template. So it's a monotonically increasing function that maps clock time in each trial to the time in the template space. So if we just allow for linear warping functions and only change the y-intercept, this induces a shift on each trial. We can relax this and change the slope as well, and this leads to a uniform stretching or compressing of the dynamics. One new technique that I introduced in my PhD was the ability to fit this model with piecewise linear warping functions, which are also very simple and interpretable from a scientific standpoint. In the most general case, you can use a nonlinear warping function, and this actually has existing work in the literature under the name of dynamic time warping. But what we found was that dynamic time warping works well for engineering questions where there's low amounts of noise and complex temporal deformations, but the amount of noise in neural data meant that these simpler models were less likely to overfit and gave us more interpretable results. So with that, I'm going to move on very quickly to questions, but just to say that this model was able to pull out oscillations in several different data sets, even though it wasn't necessarily the express purpose of that model. So with that, I'm just going to summarize and say that we can pull out really long-term trends related to learning using these methods, as well as very fine timescale, precise firing patterns. And with that, I'll take questions.